Okay, hi everybody. I'm gonna introduce myself. Tomas can't make it. He's my host actually. So everybody in the front row knows me. Everybody beyond then probably doesn't. My name is Ben Evans, and I'm from Canada. I'm at McMaster University, and I'm visiting Tomas's lab and interacting with these wonderful people in the front row. I've been here since September, and I'll be here until June. So if you want to chat with me about anything, I would love that. I think that hopefully by the end of today, you're going to know a lot more about mitochondrial and nuclear interactions and also more about macaque monkeys. I'm going to talk today about mitonuclear incompatibilities in these primates and how behavior potentially influences genome evolution and vice versa. So here's the picture of Mel, I promise, but I wanted to thank a bunch of people, past and present. So at McMaster, former graduate students, John Long and Iqbal, helped with data collection and analysis and also sample collection. Elsewhere, Don Melnick was my graduate advisor in the 90s. He since died, unfortunately, but he kind of introduced me to macaques in general. Yatna and Novi Arandiani are collaborators in Indonesia that facilitated sample collection when I was a graduate student, and also Yatna collected some of the wild-caught samples of macaques from Indonesia that I'll talk about. And then more recently, James, Jeff, Alan, Ravi, and Tomas have played roles, and also the Illumina team, Sebas, Arune, Javi, Nuria, and more recently Leah have played roles and will play roles in analyzing some of the data that we have from whole genome sequencing. And then, of course, everybody else, it's been like super fun to be here and interact with people. I've had like just an awesome time making new friends and learning about you guys' work. And then I also want to thank some funding agencies. So I'm no longer in the United States, thankfully, but I once was, and the National Science Foundation supported me as a graduate student and as a postdoc. And then my university, McMaster, and also the National Science and Engineering Research Council in Canada, and the Canadian Foundation for Innovation and the Digital Research Alliance have provided computational support. Okay, so I think probably everybody has heard of the endosymbiotic theory of the origin of mitochondrial DNA. So Dr. Margulies was the creator of this hypothesis, and it's now accepted as true. Supposedly, she submitted a paper about this idea to 15 different journals, and it was rejected from all of them. But it's now accepted as true, where mitochondrial DNA and the mitochondrial organelle was actually derived from a free-living alpha proteobacteria that was able to metabolize oxygen and produce ATP. And of course, now there's this endosymbiotic relationship between eukaryotes and mitochondrial DNA, where both the organelle and also the eukaryotic cell relies very much on an exchange between interactions between these two structures. And so in humans, we have like 25,000 genes. And in mitochondria, there's its own genome that has only 37 genes. And that's far less than the ancestral copy number, gene copy number. And that's because it's basically streamlined its genome. And the only way it's able to do what it does is through what I'll call N-interact genes, which are genes that are encoded by the nuclear genome that get transcribed and translated in the cytoplasm and then exported into the mitochondria, where they interact with mitochondrial DNA, RNA, and protein to carry out crucial functions like oxidative phosphorylation. Okay, so these N-interact genes, there's like a lot of them, so about 12,000, and they're necessary for mitochondrial function. So mitochondrial function has a lot of, I think, cool attributes that are reminiscent of its bacterial origins. So, for example, it encodes these genes that include RNA genes and tRNAs that are necessary for translating protein-coding genes and also protein-coding genes. And it actually has these polycistronic RNA transcripts that it uses to express different genes. And so this is the light strand. There's one protein-coding genes and several tRNAs. And this is the heavy strand. And so I think you guys know that these proteins get expressed, and also the RNAs get expressed and participate in mitochondrial functions, such as oxidative phosphorylation. And so this is a crucial process to generate ATP. And you can see, for example, there's 5 complexes that do this, and they are 4 out of 5 of them are encoded by both mitochondrial and nuclear proteins. So, for example, subunit 1 has 7 proteins that are encoded on the mitochondrial genome, and then 39 that are encoded by the nuclear genome, translated into cytoplasm, and then exported into the mitochondria. There's only one complex that's only nuclear, and that's complex 2. So in addition to this sort of life-sustaining function of mitochondria, it needs to be able to replicate itself. So it needs to copy its DNA, and that's done with a DNA polymerase that's also encoded by the nuclear genome and then transported into the mitochondria, and then the replication happens within the organelle. And then, of course, it needs to transcribe and translate its protein. So mitochondria has a different genetic code, and so actually it has its own set of tRNAs, and it has its own ribosomal assembly, which itself is a mosaic of proteins and RNA that are mitochondrial and nuclear encoded. So it's got a large and a small subunit that are mitochondrial encoded, and then it's got a bunch of proteins that interact with these RNAs to facilitate interactions with tRNAs and the reading of the mRNA and so on. And so all of these things are kind of crucial for life and influence essentially every phenotype in a eukaryote, because if you mess up energy production, you basically die, or you would have a massive impact on phenotypic evolution. And so we need mitochondria to generate ATP. We need it to do cell signaling and also maintain calcium homeostasis, and it's also involved with programmed cell death. And again, if you have mutations that disrupt these functions, you can have pretty severe phenotypes. So you can have reductions in copy number of mitochondrial DNA. This has been shown in humans. You can decrease the production of ATP and reduce your ability to make energy. And then that can cause the accumulation. If you sort of disrupt different stages of the electron transport chain, you can generate an excess of oxygen, a reactive oxygen species, which cause oxidative stress and toxicity to cells. And so one of the, I think, cool things about mitochondria, and sort of motivates what I'll talk about today, is this idea that mitochondrial DNA could be a so-called ticking time bomb. So mitochondrial DNA is maternally inherited and haploid. It gets passed from mother to offspring, but never through, well, in most species, not from the father. And it also doesn't recombine. And so we know from studies of evolution that recombination is necessary to decouple good and bad mutations and allow for natural selection to operate efficiently. And if you lack recombination, then we expect that phenomenon called Muller's ratchet would operate, where you have stochastic loss of the least loaded chromosome that decreases a population's fitness. And so that's what we expect from mitochondria. If it's not recombining, we expect stochastically to lose the best version of mitochondria in a population, and that ratchet will click towards a lower fitness. But so if that's the case, you might imagine that maybe mitochondrial DNA is going to kill species, because eventually it'll become so bad that those species will perish. But that's not what we see. We have a lot of lovely eukaryotes, including ourselves, that have mitochondria, and we have not suffered Muller's ratchet for some reason. And one proposed reason is that there are compensatory mutations in the nuclear genome that allow for changes in the mitochondria that could be otherwise deleterious, and basically compensate for those deleterious mutations by evolving themselves in an environment that does have recombination. So the idea of compensatory evolution to diffuse this bomb is one that I'll talk about today. Okay, so in primates in general, and macaques in particular, we have some interesting sex-specific behaviors that differently influence the distribution of geographic variation in mitochondrial and nuclear DNA because of their different modes of inheritance. So in macaques, females are sedentary. They stay in the same group that they're born in. So they live with their mothers and their grandmothers and their aunts and their daughters, and they don't leave that group over evolutionary time, over many, many generations. Males, on the other hand, are obligate migrators. So when they reach sexual maturity, they tend to disperse. And it's thought to be favored by natural selection because it avoids inbreeding. But as we'll see, it also has big implications for how mitochondrial DNA variation and nuclear DNA variation is structured over time and space, right? So if you have maternally inherited genetic elements that are only passed on by females and don't move around, you might accumulate very large differences over geographic space in mitochondrial DNA. And you would expect males are dispersing the nuclear genome to a greater degree, still affected by isolation by distance and so on, but you would expect population structure in mitochondrial DNA to be high relative to nuclear DNA because males are moving around and mixing that up. Does that make sense? Yeah. And so just looking at this in a cartoon way, imagine you have populations where the females have the mitochondria as expected, and males are joining different groups to reproduce. And in the beginning, everything's fine. Everybody's compatible. Over time, because females aren't migrating, their mitochondrial DNA will diverge, which is why I've got a little bit different shading. And then eventually you'll have these different populations that have potentially quite different mitochondrial DNA. And again, there still might be structure in nuclear DNA, but it'll be less substantial because males are moving around a lot. And so if you take a male from one population and go to an adjacent population, there may be compatible interactions between the nuclear genome of that male and the mitochondrial genome of the female that allow for reproduction and fit offspring to arise. Similarly, you might have that same male go to a more distant population. Maybe that pair can reproduce and they can produce offspring, but maybe those offspring will have sort of inefficient oxidative phosphorylation, and maybe they would have lower fitness, which is why I've got that sort of green shade there. And there might be some combinations where males go to another population and try to reproduce with a female, but their genetic variation in their nuclear genome is incompatible with the mitochondrial variation, and so you have no offspring. And so this sort of creates this behavioral difference between males and females, potentially creates a sort of interesting evolutionary arena to explore evidence for natural selection and look at how cytonuclear interactions influence fitness. And so again, the particular monkeys that I'll talk about mostly today are macaque monkeys, and they're in a broader group called papionins, which I think you've all seen examples of. So this baboon from Kenya is a papionin. This gelada from Ethiopia is a papionin. And this macaque monkey from Sulawesi is a papionin. And all of these guys, almost all papionins, not all of them, but almost all of them, have female phallopatry and obligate male migration. So we expect a lot of divergence between mitochondrial DNA in different populations, but less so in nuclear DNA. Does that make sense so far? Okay. So I want to explore kind of two projects. One's published and one's just starting. The first, well, both of them look at cytonuclear interactions, mitonuclear interactions. And the first question that I want to explore is whether we can find evidence of atypically strong natural selection on mitonuclear interactions within species of macaques. And then I want to look at the same question and ask whether we see this between species of macaques that are hybridizing. So I think you guys probably are aware of this, but natural selection influences the distribution of genetic variation in genomes. And so I've just got a simple example here where we have different chromosomes represented by lines and different mutations or dots. And let's imagine the black ones are neutral. They have no effect on fitness. The green one is beneficial. And so if that was a new mutation, over time you might expect natural selection would favor increased frequency of that beneficial mutation, potentially swamp out an entire population so that one chromosome and its linked mutations are more frequent in a population and maybe even fixed as is shown here. And so in general, we expect natural selection affects the distribution of genetic variation in populations, for example, by introducing stretches of homozygosity on either side of a beneficial mutation in the case of positive selection, which is shown here. So we expect what we call runs of homozygosity to be longer in areas that are under positive selection. We expect that there might be differentiation between different populations in areas that are under natural selection, where maybe local adaptation changes the allele frequencies in different populations. And then also, obviously, we expect changes in polymorphism, both in positive and negative selection, depending on the nature of natural selection. And so I've just shown here positive selection, but obviously there's lots of different flavors of natural selection. Balancing selection is an exception where you'd have increased diversity in a population, but that's not something that I'll talk about today. So in humans, we can see lovely examples of this, which is kind of cool, even though humans have a relatively small effective population size. And this is just a graph that shows runs of homozygosity on either side of an allele that confers persistent expression of lactase, which allows us to metabolize dairy from domestic animals. And you can see that it was favored by natural selection. This mutation, or different mutations, evolved multiple times in humans, but you can see in red are these sort of stretches of homozygosity that flank this beneficial mutation in these people that had lactase persistence. And then the individuals that didn't have lactase persistence have smaller tracts of homozygosity because they had the ancestral allele that wasn't beneficial. So natural selection came through this population, and some individuals that had the beneficial allele, it was favored and increased in frequency and sort of erased the variation on either side. And so these runs of homozygosity are one metric of many that one can use to evaluate evidence of natural selection. Well, one of the problems of that approach is that there are other sort of neutral phenomena that influence diversity as well. And this is from a review article by a guy that actually gave a talk here like 3 weeks ago or 4 weeks ago, Dr. Caballos. And it shows that changes in population size can also increase or decrease runs of homozygosity in interesting ways. So on this side, for example, you can see that the number of runs of homozygosity in individuals, which are indicated by dots, and also the summed length of runs of homozygosity in these individuals are small in a large population relative to a population that's been bottlenecked, where those runs both increase in number and also increase in summed length after a bottleneck. Okay, so the problem that we have is how do we distinguish between demographic effects, which we're kind of not really interested in, and natural selection effects, which is what we care about. And the way that I'm doing this is to look at signatures of natural selection in the same individuals for different categories of genes. So we got this category of N-interact genes that we're hypothesizing should be under natural selection, and I'm going to compare genomic metrics of those genes to other genes in the genome that are not involved with cytonuclear interactions. Okay, so that way we can kind of control for demographic effects because demography usually affects an entire genome, whereas natural selection usually affects a subset. Does that make sense? Okay. Yeah.
Do you expect any interactions to be under more natural selection than the others?
Yes.
That's not.
Yeah, that's what I
expect. What the more? I understand that they would be under natural selection, but the rest of the genome as well, to some extent. How do you know that they're going to be more?
I don't know for sure, but I'm hypothesizing because the speculation is that the interactions that involve mitochondrial DNA should be atypically important. So other genes that are involved with height or hair color or eye color or something like that, I think are important, but not as important. So it's a relativistic speculation that is testable. Okay, so I'm sort of maybe tilting the scale a little bit because I'm not going to look at all 1,200 N-interact genes that we know get exported to the mitochondrial organelle. I'm going to focus in particular on about 200 autosomally encoded genes that I know from other studies have direct interactions with mitochondrial DNA, RNA, or protein. Okay, so it's these genes in particular that I expect natural selection to be atypically strong because they have these direct interactions with a really rapidly evolving set of molecules. And I should mention as well that mitochondrial DNA in some primates evolves about 20 times faster than nuclear DNA, so we expect it to be really require compensatory evolution in order for it to function. Okay? Okay, so I'm going to tell you about sort of two suites of data. The first one's published, and it includes 4 data sets from 3 species, and the second one's unpublished. And so the first data set I'll talk about includes 2 species that have what's called mitonuclear phylogenetic discordance, and I'll explain what I mean by that in a moment. And it includes the stump-tailed macaque, Macaque arctoides, and a subspecies of the long-tailed macaque, Macaque vesicularis aurea. And then I'm going to focus on 2 other data sets from whole genome sequences from the rhesus macaque. One's captive and one's wild caught. And these were all previously published data sets. And so when I say mitonuclear phylogenomic discordance, all I mean is that these species that are vesicularis aurea and arctoides have strongly supported mitochondrial phylogenies that disagree with the nuclear phylogeny that's also strong. Okay, so in this case, species B and C have closely related mitochondria, but A and B have closely related nuclear gene. And so that is the case in macaque arctoides, where mitochondrial DNA of arctoides is closely related to the rhesus macaque, but nuclear DNA is closely related to the toque macaque from Sri Lanka. Okay, and that's again been established by other people. It's very well supported. We have whole genome data that support this for the mitochondria and the autosomes. The case in this subspecies of long-tailed macaque is even weirder, I think, because this subspecies, which occurs along the coast of Burma and the western coast of Thailand, this one little pocket of this long-tailed macaque, which has a wide distribution, has mitochondrial DNA that's clearly derived from Sri Lanka, from the toque macaque. But the mitochondrial DNA from the rest of the species throughout Indonesia is very diverged. But when you look at the nuclear genome, not surprisingly based on their appearance, this subspecies has close relatives in the other subspecies of long-tailed macaque. So it appears that mitochondrial DNA was kind of introgressed or inherited by the subspecies only, but not throughout the whole species. And you can imagine by introducing really diverged mitochondrial DNA, the question is whether the nuclear genome has to react to that and undergo compensatory evolution to allow for mitochondrial function. And then the other data set I'll talk about is the rhesus macaques. And this species is interesting because rhesus macaques are the most widely distributed non-human primate. So they occur from Pakistan all the way over to East China, and from central China down into like northern Thailand and northern Vietnam and so on. So they have this massive range, bigger than any other primate other than humans. And as we'll see, they also have really, really diverged and diverse mitochondrial DNA. So again, you can imagine males moving around this massive range. They're going to go to these new groups that have mitochondrial DNA that's really diverged, and potentially that's a shock to their genome that involves natural selection in order for their offspring to be able to reproduce or to live. And so what we've done first was to take whole genome sequences and assemble new mitochondrial genomes from a bunch of these samples and then combine it with previously assembled mitochondrial genomes. And we generated this time-calibrated phylogeny that shows some of the remarkable diversity of macaque mitochondrial genomes in the focal group, but also in other groups as well. So I'll highlight a few things. One is that Macaca arctoides, again, has this close mitochondrial affinity with Mulata that stretches back about 4 million years, but actually its nuclear genome is more closely related to Seneca. Okay, so it's like 5 million years diverged. Similarly, Macaca fascicularis aurea has close mitochondrial DNA to Seneca, but it's actually nuclear DNA is very closely related to fascicularis, the other subspecies. So again, the mitochondrial genome is, roughly 5 million years diverged from the nuclear genome, the common ancestor with the nuclear genome. So it's really, really diverged. It almost would be like a human having chimpanzee mitochondria. So that would be bizarre. And then, of course, in Mulata and the rhesus macaque, we have really diverged lineages within this species that far, far exceed what we see, for example, between chimpanzees and bonobos. So the time of coalescence for rhesus mitochondrial DNA is greater than what we see amongst species of chimpanzee within one species. Okay, so we have a lot of variation in mitochondrial DNA, and these numbers just show the number of individuals in each sample. So we expected to see evidence of natural selection on N-interact genes in these different species, and that's what we found. So I'll just show you some different lines of evidence that support this. So, for example, if you tabulate runs of homozygosity, the length of them, in runs of homozygosity that contain N-interact genes and those that don't, and then compare that to other parts of the genome that have no genes at all, you'll find that in general, runs of homozygosity that have N-interact genes tend to be much longer than those that don't, and that includes those that don't and have no genes, or those that have other genes that don't get direct interactions with mitochondrial DNA. So this is consistent with positive selection, where you have much more homozygosity stretching on either side of these regions that have proteins that get exported and interact directly with mitochondrial and nuclear DNA. Does that make sense? And so one might say that maybe this is because the regions that have these nuclear encoded proteins that get exported to mitochondrial DNA are also regions that just have a lot of other genes. So there's high gene density that is explaining this. It's not actually selection on these genes that get exported to the mitochondria. And so to explore that possibility, we use a linear model, which basically has a binary variable that's either 1 or 0, depending on whether a run of homozygosity does or does not have an N-interact gene. And then we have the number of genes that that run actually includes. And so we're going to relate these two variables to the long transformed run of homozygosity length. what I'm going to show you is plots that show the predicted relationships for runs that do and do not have and interact genes over what I would argue are biologically realistic numbers of genes that encompass basically 90% of the distribution of the data. So I'm not going to show the points because there's 26,000 points or something, but I'm going to show you the fitted values for the model. And what we see consistently is that, as expected, the runs of homozygosity that have N-interact genes are far longer, no matter how many genes are in those runs, than those that don't have N-interact genes. And this includes only the runs that have other genes. It doesn't include the runs that have no genes at all. So it's a sort of apples to apples comparison. And again, it's very consistent with this expectation that we have. And that was true not only for macaque arctoides, which I just showed you, but also for basically all of the species that we looked at. And so what this means is that 55% of the points had only one gene in those runs. And for that particular set of the data, the runs that had an interact genes were very strong. And so I guess it was surprising to me that I didn't see as big of a difference in the fascicularis data set, but it was still significant over biologically relevant aspects of the gene density. This was one that was sort of overlapping confidence intervals. But in general, we saw, something that was consistent. Yeah.
Can I ask first the background that you compared to? Is it also controlled for the fact that it has direct interactions with tRNAs or RNAs or other proteins? Because the restricted set of genes that you look at is not just any set of genes, right?
No, it's not at all. Yeah, so in that restricted set, I included proteins from the nuclear genome that do interact with tRNAs. So is that what you mean? So the nuclear encoded genes that catalyze the charging of tRNAs. I think there's three categories. The other one is nuclear encoded genes that interact with the mitochondrial ribosome to form the ribosome, and the other one was nuclear encoded genes that are involved with DNA polymerase.
If you apply the same set of constraints to the other nuclear genes, do you think that would change things?
That's a good idea. So I haven't done that comparison. So I see what you're saying. So I could take, for example, the nuclear encoded genes that catalyze the charging of tRNAs for the cytoplasm and compare them to. That's a great idea. I haven't done that.
And then, do you understand why their slope is not the same in these problems?
Well, there are different data sets for each species, and
it's in the blue and red.
Right. So that's because there's an interaction term, and so what it means is that the relationship between gene density and the length of the amount of homozygosity depends on whether it has an interact or a non-interact gene. And so, for example, in this species, the more genes there are in that run, and that run has a stronger positive relationship if you don't have an N-interact gene. It's a subtlety I don't know why. Yes. It's a subtlety in the statistic, and that's why I've only plotted the 90th percentiles to show sort of biologically relevant aspects of it. I mean, if you carry this forward, then there'd be a point where the non-N-interact runs of homozygosity would be predicted to be larger, but there would actually be none of those in the data. Does that make sense? Any other questions? And so we can do other metrics of natural selection. This is FST, which again we predicted to be more different for N-interact. This is a genomic windows analysis. They're 30 kb windows. And we see a subset, at least, of the N-interact genes having atypically high population structure in pairwise comparisons, in this case, that involve the subspecies of long-tailed macaque. And I'm not going to show you all the data, but in general, we find a peak in at least a subset of the N-interact genes. In FST, we tend to find lower polymorphism, which is consistent with the runs of homozygosity. We also looked at protein evolution, which tends to be faster, which is consistent with compensatory evolution. We also looked at Tajima's D, which is a metric that's influenced both by natural selection but also demography. And we generally expected Tajima's D to be more negative in N-interact windows than in non-N-interact windows, and that's generally what we found. But the signal I'd say was strongest for FST and runs of homozygosity. Okay, so far, I think the data would suggest that Natural selection on this subset of genes seems to be atypically strong relative to other genes in the macaque genomes. And that influences the amount of polymorphism in flanking regions, for example, by increasing runs of homozygosity. And then I'll discuss kind of broader sort of whole organism implications of this later on. Does that make sense so far? Okay, so now I want to talk about a new data set that looks at gene flow mostly between species. And so I think many of you know that gene flow can be adaptive. And so this has been postulated, for example, with Neanderthal-human interactions. Here's a wonderful example from humans from Madagascar. So the Malagasy people are an admixed group that has Asian and African ancestry. And in general, if you scan their genomes of lots of people from Madagascar, you'll see that their Asian ancestry is roughly 40%. But there's one point on chromosome 1 where the Asian ancestry plummets. It's almost very low, 0 almost, or below 10%. And there's a lot of African ancestry in that part of the genome. And that particular genomic region has the Duffy blood group allele, which confers malaria resistance. So I think it's a wonderful example from humans where genetic exchange allowed natural selection to have targets to choose and get rid of. And clearly it shows the allele that had obvious phenotypic benefit of conferring malaria resistance. And so by looking at variation in ancestry, we can learn about how natural selection is operating and what it's favoring or disfavoring. And so we're going to go to a different part of the range of macaques now. This is Southeast Asia, mostly the country of Indonesia. You guys probably have heard of Wallace's line. Is it everybody else? Okay. No? Okay. So Wallace is a kind of underappreciated figure in evolutionary biology who corresponded with Darwin in the mid-19th century. He lived in Indonesia and he traveled and lived in this area. And he noted, for example, when he went to the forests of Sumatra and Borneo, he saw things like orangutans and leaf monkeys and rhinos and elephants. But then when he went to the island of Sulawesi, he didn't see any of those things. And when he carried on through to Papua New Guinea and Australia, he saw lots of marsupials like kangaroos and wombats and things. And he only saw one in Sulawesi and none in Western Indonesia. And so this is, he noted, it was one of the most precipitous faunal exchange or faunal turnover events in the world, basically for terrestrial fauna. And he didn't call it Wallace's line, so that'd be kind of narcissistic, but we call it Wallace's line. It's meant to demarcate the transition zone between the Asian and Australian faunal zone that occurs in central Indonesia. And we actually know now that there's a geological basis for this because Borneo, Sumatra, and Java were connected to mainland Asia by land during the Pleistocene when the global sea level was lower because there was ice caps. And Sulawesi was still isolated. New Guinea and Australia were connected by land as well. And so what's interesting about this area from the perspective of macaques is that unlike many other large-bodied mammals in this region, macaque monkeys made it into these Sunda Islands, we call them, but they also managed to transcend Wallace's line to get to Sulawesi. And when they did, they underwent this explosive diversification to give rise to 8 different species. So there's, I think, 23 species of macaques, and a third of them occur on this tiny island that's about 4% of the range. So macaques occur from Morocco to Japan, and again, from China down into Sulawesi, and a third of the diversity is on this tiny island. And as well, there are these little areas that there's admixture between adjacent species. So these different species you'll see are actually distinctive genetically as well. They have obvious differences in fur and in the shape of the ischioclosities and their body size and behavior, all sorts of really interesting things that again sort of underscore the diversity on this amazing island. So Sulawesi is cool for lots of other reasons too. Anyway, so we've got this diversity, and as a graduate student, I still look back on this like one of the funnest things I've done in my life. I was able to go to Indonesia and work with colleagues from the University of Indonesia and travel around Indonesia, and especially Sulawesi, and take advantage of the fact that people there keep these monkeys as pets. And so I could go into a town, roll down the window, and say, "Ada yang kaliharan monyet di sini?" And they'd look at me strangely, and they'd be like, "Yes, right over there." And so I could go over, and I could use ketamine they let me use ketamine in the 90s, so I would anesthetize the animals and draw blood. It's crazy. Who could do that now? Anyway, so, and the great thing about this situation is that in many cases, the people that owned the animals knew where it came from. So they either caught it themselves or their friend caught it, and, it was from the forest over there. And so I could get really good metadata associated with all the animals, and then I could confirm their species identity just by looking at them. As well, because they were anesthetized, I could kind of do sort of routine or basic first aid on them. So some of them were in pretty bad conditions, and so I was able to kind of help the animal out. So in my view, this is a sort of ethical way to access samples that minimally impacted these animals in the wild. And so what we ended up with over the years was, over 162 samples, but we've sequenced now 162 of these genomes. And so this just shows the distribution of some of them. It includes pig-tailed macaques from Sumatra and Borneo, Subaru, these are from zoos actually, macaque Subaru from Mentawai Islands, and then a bunch of these samples that were mostly collected from pets. But as we'll see, there's also wild-caught samples that Yatna Suprayatna collected as a graduate student in the 80s. And so we have not shown here are 44 hybrids. Most of them are from this hybrid zone between Macaque Mara in the southwest and Macaque Tonkiana in the central part of Sulawesi. Okay, so we have this sort of really, I think, unusual and exciting genomic resource to look at. And then we also have an outgroup of the toque macaque. And so one of the first things that we did with the early stages of the whole genome sequencing was, again, to generate whole mitochondrial genomes from these guys and estimate a phylogeny that was time calibrated. And, we found shocking levels of diversity on this island, again, which is such a small component of the total range. So we see similar levels of diversity and divergence between Sulawesi macaques that we see in baboons throughout all of Africa. So, I mean, it's, a tiny range, and we're seeing really extraordinary variation. Similarly, we see about the same level or a little bit more diversity in the Sulawesi macaques as we see between the different species of chimpanzee. When you translate peptides that are mitochondrially encoded, on Sulawesi macaques, you also see a level of non-synonymous divergence or protein differences among Sulawesi macaques that's about the same as what we see between humans and chimpanzees. And so the mitochondrial DNA of these species is really extraordinarily diverged despite their sort of close geographical proximity. And I'll just go back. So just by way of scale, that's about 100 kilometers. So this is a really small sort of, you can drive, especially now that the roads are better, you can drive from here to here in 24 hours. So it's like incredible concentration of primate diversity on this tiny island. Does that make sense so far? Okay. The other kind of fun thing that we would see, and that we see many times in the new data, is that there oftentimes were species that were clearly identifiable as belonging to one species, but they had mitochondrial genomes from another. So here's a macaque tonkiana. It looks like macaque tonkiana in every way, but it's got this diverged mitochondrial lineage from macaque hecki, suggesting that it's hybrid, or that there's some admixture of mitochondrial DNA going on. And so we began by doing some sanity checks. So do we see in a principal components analysis things sort of sorting as we would expect they would, and they do. So we see the pigtailed macaques and macaque Subaru that were on the western part of Indonesia being separated on the first principal component from the Sulawesi macaques. If I look only at Sulawesi, we can again see geographic structure to the plot. So these guys are from the northern peninsula, which we know from previous analyses are closely related. And then this is from the west of Sulawesi. And then excitingly, we see these guys that are intermediate that are, we know to be now hybrids between macaque tonkiana and macaque mora. And these are wild-caught samples that were sampled by Yatna Suprayatna in the hybrid zone between these guys. And so everything sort of looks the way we would hope. Another thing people often do is do admixture analyses, and we did that as well. And so here again, you can see, for example, these hybrids, which I divided into two groups, one that has Mora mitochondria and then one that has Tonkiana mitochondria. So these are hybrids from these two parental species. You can see that they're kind of admixed genomes. So they appear to be formed from both of the parental species. And we see other kind of soothing things like macaque nemostrina from Sumatra is different from macaque nemostrina from Borneo, and other things that you kind of expect. So, not sort of earth-shattering, but comforting. And I want to focus now again on these these two species from southwestern Sulawesi. This actually shows some of the variation that you see in estrus swelling. So females have this really remarkable species-specific estrus swelling that looks very different between the species and is cyclical with their cycle. And there's also kind of interesting differences in body size. Tonkiana tends to be bigger than Mara. Males are always bigger than females in all species. And so let's look at the first mitochondrial DNA. This is published, maybe some of you were born around then. This was published, one of the first studies for my PhD work. And I just used Sanger sequencing to look at variation between individuals in the hybrid zone and in the parental species. And in the hybrid zone, which is this low-lying area that's periodically inundated with water, there's a really sharp boundary between mitochondrial lineages in the north and the south. And so there's two populations where there's shared lineages. This is a male individual that went to that population and is going to transmit it. These are 6 individuals, and these are 3 males and 3 females. So these 3 males have been migrated north. And so this boundary between mitochondrial lineages is exactly what we'd expect if females are phylopatric. So we can now sort of go in further and take these hybrid individuals, which, as you'll see, we know are hybrid, and we can try to identify admixture blocks. So regions of these genomes that are derived from each of the parental species. And so to accomplish this, I used a program called AdmixFrog, which implements a hidden Markov model. And the idea here is that you've got hidden and observed states. So the observed states are genotypes across the genome, and the hidden states are the extent of blocks of the genome that are inherited from one or the other parental species. And so this is a nice program as well because it allows for heterozygous hidden states. So you can infer not only homozygous Mara regions, but also heterozygous Mara Tonkiana regions. And so that can vary by individual, obviously, because they all have a separate evolutionary history. And so what you end up having inferred are introgression blocks for each of these hybrid individuals where you can say, okay, that's from one parental species or another, or it's heterozygous. So now that we've got the genomes dissected out, I can begin to ask sort of predictable questions, which would include, for example, if I look at individuals that have Mara mitochondria, is it the case that introgression blocks that carry and interact genes tend to be derived from that species? In other words, is natural selection favoring an enrichment of favorable or compatible interactions between genetic variation in the nuclear genome that's had a longer period of coevolutionary history with the mitochondria? That makes sense? That's a prediction going in, and you can test that, relative to a null expectation where there's just sort of random assortment of introgression blocks throughout the genome. And so this is like a huge amount of data that I put on one slide, but it's fairly easy to digest. So rhesus macaques have 20 chromosomes, and each of the columns represents a chromosome. And then I've got individuals from the Mara parental species in rows. These are individuals from the Tonkiana parental species. And then these are hybrid individuals that have Mara mitochondrial DNA. And these are hybrid individuals that have Tonkiana mitochondrial DNA. And you can see that there's an inference of a lot of admixture in these individuals. It's not like just, some huge section gets a little bit of DNA and then another one gets from one species and then there's another region from another. It's sort of really small expression blocks that are scattered throughout the genome, which is suggestive of a lot of sort of backcrossing of these individuals. And so again, the question that I want to ask now is if we go in to individual regions that are introgressed in these hybrid individuals, do I see and interact genes in the autosomes that match their mitochondrial genomes? The prediction is yes. And of course, we'll see some, but do I see an over-enrichment of them? And the answer on one level is a little bit mealy-mouthed. So when I do this, I develop a permutation test, which basically scatters the gene identity across the introgression blocks, and then looks at, relative to those permutations, whether the observed distribution on introgression blocks is higher than what you'd expect by chance. And I find that 6 out of 44 are individually significant. And when you do multiple corrections, it's only 2. So it's not really a strong signal based on that permutation test. But when you combine probabilities using Fisher's method, it does turn out to be significant. But I think what this tells us is that across all of those genes, the signal is not particularly strong of enrichment in hybrid zones. But as we'll see, across individual genes, it actually is. And so the question that we can then ask, well, first of all, does that make sense so far? Yeah, more or less? Okay. So we've got 199 and interact genes that we know whether they are or are not on integration blocks in 44 hybrid individuals. So I can now say for each of the interact genes, are they disproportionately found on integration blocks that match the origin of the mitochondrial DNA or not? And so I calculated that, and I'll sort of walk you through this. We've got a maximum of 44 individuals. If we allow for integration blocks to be either homozygous or heterozygous, there were 6 N-interact genes that were found on integration blocks that match their mitochondrial DNA in all 44 hybrid individuals. If we focus only on homozygous integration blocks, there was 1 interact gene that was found in all 44 individuals in homozygous integration block, and there was 1 that was found in 38 out of 44 of the hybrid individuals. So I think what these graphs show us is that there are some N-interact genes that are atypically prevalent in homozygous or heterozygous integration blocks that match the origin of their parental species. Does that make sense? I think it's a strong effect. I was pretty excited by that. And so what are those genes? These are the names. So they are mitochondrial ribonucleoprotein in the large subunit, and also in the small subunit. There's 4 of these and 1 of those. And then there's also a member of the oxphos complex 1. And so one might say, well, maybe these genes are all linked, and this is actually just natural selection for 1 region. And so that turns out to not really be the case. So all of these are on separate chromosomes completely, so they're completely independently being selected on. DMAC2 is on the same chromosome as MPRL4, about 30,000 base pairs away. So it's possible that linkage could influence those 2 genes together, but all the other ones are completely independent events. Does that make sense? And so, again, 4 of these are in the large subunit of the ribosome. 1 of them is in the small subunit, and then 1 of them is in complex 1 of phosphogenic phosphorylation pathway. And so that was exciting for me, but we can burrow down a little bit farther. Last year, there was a paper published that has the crystal structure of the human mitochondrial ribosome that includes RNA, which is in gray, and all the proteins. And so there's different meanings of the colors, so I won't explain it, but we have a really good idea about protein RNA interactions in the human mitochondrial ribosome. And we can look at, for example, these genes and ask where in the mitochondrial ribosomal RNA do these proteins actually interface. And so here's a crystal structure of MRP1L in humans, and you can see this gray stuff. It's a little bit hard to understand, but this is a stem loop region of the human mitochondrial ribosomal RNA. And this is actually the secondary structure of that. You can see there's Watson-Crick base pairs that form stems, and then little non-Watson-Crick base pairs that form loops. And this protein interacts directly with these particular RNA residues. And so we can then say, well, are these actually variable in the macaques we're looking at? One would hope they would be, because otherwise, what have we found? And of course, it turns out they are, which is really neat. But as well, it appears that the secondary structure of this particular stem loop section in humans is actually quite different in the macaque monkeys. And so one of the things I'm sort of eager to do as a next step is to use artificial intelligence to try to develop secondary and tertiary structures of these ribosomal RNA sequences from macaques and look at how these regions are actually evolving in sections that are influenced, that interact directly with these proteins of interest. Okay, does that make sense so far? Is anybody tired yet? Okay. You won't admit it, you're too nice. Okay, so I'm going to try to now sort of step back and talk about why this matters. And I think it matters for a lot of reasons, and they actually affect humans. So one reason is that I think cytonuclear or mitonuclear interactions potentially influence and catalyze speciation through Jobzansky-Muller interactions. And so you guys may know what this is. This shows basically incompatibilities arising between different autosomal loci that acquire mutations that are incompatible across species. And so that's the basis of the Dobzhansky and other interaction, is that it evolves out of spaces. And so imagine you have two genes. One's got little a's on it, and one's got little b's. In one population, you have a mutation that fixes in the B gene, and then in that same population, you have a mutation that fixes in the A gene. And let's say that big A has never interacted now through evolution with a little b, right? It's always been separated because the big B fixed first. And so now if you make a hybrid between the big A, big B, and the little a, little b, you end up with a heterozygous individual where there's potentially a dominant negative interaction between the big A allele and the little b. Okay, so this is actually a solution to a big problem in speciation genetics, which is how can you get incompatibilities within a locus if you have to go through a heterozygous phase? And the answer is that you have interactions between loci. But you can imagine I could draw this figure not as pretty, but equally well with cytonuclear interactions, where there's incompatibilities that arise in the autosomes and the mitochondria that cause species incompatibilities when you make hybrids. So, and it's no surprise, therefore, that macaques are one of the most diverse primate genera that we know of. Another kind of crazy thing is that mitonuclear interactions could influence sexual dimorphism in interesting ways. And so papinins in general have really sexually dimorphic males and females. Mandrills, the males are 3 times as big as females. You imagine if the males in humans were 3 times as big as females, it'd be a nightmare, right? So, these guys have really big differences in canine length also between the sexes. As well in macaques, there's really big differences in estrus swelling. And so this is known to be different among species and also variable within species. And it's been postulated that this could actually essentially be a signal for the efficiency of oxidative phosphorylation and also compatibility of mitonuclear interactions within and between species. So that's been proposed. One of the other, and I'm not just trying to show pictures of genitals here, but one of the other interesting observations that's potentially correlative, but also potentially causative, is that one of those species that I mentioned earlier, Macaque arctoides, turns out to have really bizarre genitals. So macaques are divided into species groups based on their genital morphology. Macaque arctoides is a monotypic species in its group because it's got a really long penis and a correspondingly long female reproductive tract. And so it could be that this particular species, which is one of the sort of classic examples of mitonuclear discordance, has these really different genitals because of cytonuclear interactions. And this is this sort of mechanism to sort of signal mitonuclear incompatibilities. So again, I don't have evidence for that, but it's sort of an interesting thing that's out there. And I just got a few more things before I release you guys, but I feel like I'm teaching a class a little bit. But one of the other sort of relevant factors that impinges on mitonuclear discordance on a phylogenetic level is what we call Darwin's corollary to Haldane's rule. So I don't know if you guys have heard of Haldane's rule, but that's where, I won't get into it. It's related to sex chromosomes and hybrid incompatibilities, but Darwin's corollary to Haldane's rule describes situations where reciprocal crosses can have different fitness effects. So in this case, if species 1, if you take a female and cross it with species 2 male, you get offspring that are hybrid and fine. But if you take a female from species 2, oh, sorry, I should have relabeled it. The female is from species 2 and cross it to a male, so it's the reciprocal cross, the hybrids are infertile. So that's an observation that we see in nature. And you can imagine if that were to happen, it could allow for mitochondrial integration from one species to another. That could create phylogenomic discordance between mitochondrial and nuclear DNA. It's also possible, even without hybrid fitness, that or hybrid incompatibilities, that there could just be higher fitness in one mitochondrial lineage that allows it to swamp through another species, even if gene flow were relatively rare or female migration were relatively rare. And in fact, we see this in humans. So I don't know if you guys know, it's actually from Spain that we realize this. We now obviously have the genomes from Neanderthals and Denisovans in humans, obviously, and there's a fossil from Cima de Huesos in northern Spain that they managed to get mitochondrial DNA and autosomal DNA and show that Neanderthal was a Neanderthal that was closely related to Denisovans, and its mitochondria and nuclear DNA were too. But more recent Neanderthals actually have mitochondrial DNA that's clearly derived from modern humans, even though their nuclear genome is not. And so we have phylogenomic, whatever I called it, we have it in humans, right? It's amazing. So we have this sort of disparity that happened through time. Why did it happen? Probably something related to, I mean, it could be anything. It could be neutral, but it could also be related to higher fitness of the mitochondrial lineage and mitochondrial DNA compared to the Neanderthals, which had a small effective population size and so on. There's lots of like kind of cool implications in humans. I'll just show you one last one. And that is this idea of mother's curse. So one of the features of mitochondrial DNA is that it's maternally inherited, which means that it doesn't have a kind of opportunity for male detriment mutations to be removed by natural selection. So if mitochondrial DNA is only being passed on from mother to daughter and son, but the sons never get a chance to pass it on, then natural selection can't really remove mutations that affect males but not females. Okay? And so these male detriment mutations potentially could accumulate in mitochondria and affect males, but not affect females, where natural selection can act. And so we see evidence of this. So in macaques, there's this really sharp female bias sex ratio in adults. So populations in the wild can be like 7 times more females than males, which is crazy. We have diseases in humans like this one, Leber's hereditary optic neuropathy, which is 3-fold higher penetrance in males than in females. It's probably also affected by the estrogen, probably has a protective effect. So it appears that this is a mitochondrial mutation that's causing a condition. Actually, estrogen is used to treat it in males, and it appears to be a male detriment mutation that disproportionately affects males. They've looked at sperm motility in different human mitochondrial haplogroups and found that there's variation, and some haplogroups are associated with lower sperm motility in males, no effect in females. And there are also cool examples in fruit flies where you can take the same genetic background and introduce natural mitochondrial genomes that are found throughout the world, and there's male-specific effects where it causes decreased longevity in males, depending on the haplogroup. And so there's all sorts of cool examples where mitochondrial DNA has sort of different genomic effects in the sexes and potentially whole organism implications. Does that make sense? Okay, so that's all I wanted to say. Thank you for listening, and I'm happy to answer questions.